the topic of stratified squamous epithelium. Now we will come up with the stratified cuboidal epithelium. In order to identify the stratified cuboidal epithelium, we need to be clarified that the length and the width of the cells of the stratified cuboidal epithelium are same. How is it so? To make the cube, as all of you knows that the length and width of the cube is same. See over here, length and width. I know it's little bit different, but we'll consider it as same. But in that of stratified columnar, you can see there is a noticeable change in the length and in the width. Length is slightly more than the width. So the example of stratified cuboidal epithelium are to be found on the larger ducts that are exocrine glands of the pancreas and salivary glands, not involved in absorptive or secretory activity. They are only lining it, not secreting the substances. Now coming to the stratified columnar, as we have discussed, the length is greater while the width is less. Length is greater while the width is less. So they lines the conjunctiva of the eye and male urethra. It's very, you can say, limited. Now coming to our most special epithelium, that is transitional epithelium. That's the most loved epithelium. So coming back to our chief guest, that is transitional epithelium, also known as urothelium, is the most important type of epithelium. You can say its most asked questions come from the transitional epithelium in the MCQs or BCQs. So we also name as urothelium, and it's the specialized type of stratified epithelium. It's nothing else but it's a type of specialized stratified epithelium. Why it's specialized and why it is not characterized in the stratified epithelium, we will discuss this. Hence the lower urinary cavity, and due to this reason this is known as urothelium. We have discussed it. It is known as transitional epithelium as it undergoes changes in undistended state and distended state. What do you mean by that? That is the histological features, that when the transitional epithelium, when you look at take the example of balloon. When you look at the balloon which is not filled with air, that is in undistended state, it appears to be small. But when you fill it up with the air, it's stretched without getting burst and without leaking air, and appears to be more bigger. That is because of its elasticity, its flexibility. Same goes with the what we call transitional epithelium or urothelium. It consists of 6 layers when it is in undistended state. You all know, because of its elasticity, when it get filled, that is in distended state, it loses its not actually loses, the layers appears to be less, that is either 3 or either 2. Then will come up, the epithelium consists of cuboidal cells over the basal layer are present of polygonal cells. That means the lowermost layer consists of cuboidal cells, that is this one. Basal cells are cuboidal cells, and then we come up with the polygonal cells, that is, I know I'm drawing it wrong way, but I can help you out with this. The most superficial layer of epithelium appears to be consist of very large dome-shaped cells. What do you mean by dome-shaped cells? It's the most important feature of this cell. It's most likely to be asked in the questions, that which cells consist of the dome cells, or the, you can call it, okay, transitional epithelium is characterized due to the presence of which of the unique type of cells. These type of cells are known as umbrella cells, as they appears to be in umbrella shape, like you can say like this. I know I've drawn it wrong, sorry. And most of them contains 2 nuclei. You have to remember it, that most of them contains 2 nuclei. It's the most important thing to be remembered. Okay, I can see if I have a picture of this. Yeah, there you go. They are the dome-shaped cells. See, this one doesn't contains 2 nuclei, but this one contains 2 nuclei, and this is characterized to be that they are the transitional epithelium. Then we have plaque. They contains plaque. When we view them under the electron microscope, the apical surface of the most superficial cells has thick and rigid areas called plaque. These plaque help make the surface resistant to toxic effect of urine. Now consider this as your transitional epithelium. The picture is very blurry. The plaque is this region, this one. This is most tightly packed region. It does not allow the toxic substances like urine to get reabsorbed, and causes difficult problems and other situations which can lead to worse condition. So for that reason, plaques are present. It's also the most important feature of the transitional epithelium. It can be asked that where is plaque present, then you will name that transitional epithelium. The interplaque region is the area between the plaques in the superficial cells of transitional epithelium. You can see over here. If these regions are not present, then what will happen? If these regions are not present in the plaque, we have studied that plaque is a thick and rigid area, then transitional epithelium would not be able to get its elasticity. It's written over here, plaque alone would make the epithelium too stiff and unable to stretch. Then what's the reason to call it a transitional epithelium? Now we'll come up with the functions. It prevents the diffusion of urine back into the tissues. We have studied in the plaque region that due to the presence of the plaque, they do not allow the toxic substances to get absorbed in the blood stream. They forms the impermeable barrier, which prevents the urine from passing into the epithelium or beyond into the underlying tissues to prevent the worst conditions. They contains, how do they form the impermeable barrier? By the presence of desmosomes and zonula occludens, which we'll discuss. They are type of a cell junctions. We'll discuss it in another video. These are the examples of the transitional epithelium. You can go through it. You have to remember it. They are very important. They are asked in the question MCQs. Then you'll come up with the apical surface. Consider this as your epithelial cell. Okay. Now you'll consider what are the different surfaces of the epithelial cells. We have discussed the apical surface. This is the apical surface of the epithelium. The uppermost, or you can say superficial layer of the, not superficial, the uppermost layer of the epithelium. And on the both of the sides of the epithelium lies a lateral domain or lateral surface. This domain is also equally important as this contains cell junctions, as we have discussed. They are all the cell junctions mentioned over here. All the cell junctions mentioned over here, which makes them impermeable, or you can say, as you have discussed in transitional epithelium, these cell junctions make them impermeable, the layer made of the cells, that is, so avoid the absorption of the toxic substances. Now coming up with the basal domain. This basal domain lies in contact with the basement membrane. Here you see there is a basement membrane or basal lamina. They contains the infoldings of plasma lemma. This is the important character of the basal domain. In order to characterize them, we have to consider that as the you can say identification point. And they contains hemidesmosomes, which is another type of the cell junction. Now coming back to our apical domain. This apical domain, you can see there are certain modifications present. I haven't discussed it yet. I have discussed the cell junctions, but I haven't discussed it yet. What are these enfoldings called? We have discussed that enfoldings are also present in the basal domain, but they are different from the enfoldings of the apical domain. We have named them as individual character. You can see this small little enfoldings. These are called villi, microvilli, while the large enfoldings, or that are this one and this one present here, are known as cilium or cilia. One is mentioned as gynoecilia, another is mentioned as stereocilia. What is the difference? We'll discuss it now. First, we'll go with the microvilli. So microvilli is the extension of the cytoplasm, appearing to be as finger-like. As we have discussed, what's the importance? As we have discussed in the intestine, they are present in order to increase the surface area and so that absorption can be more efficient. Okay, now coming up with the definition, the actual word-to-word definition. These are small cylinder finger-like projection found on the free surface of the epithelial cells. Microvilli are defined as the cytoplasmic extension of plasma membrane found on the apical surface of the epithelial cells. Now we will discuss the structure of the microvilli. Central bundle of 20 to 30 microfilaments, actin filaments, that is, they contains microfilaments that are specifically actin filaments, which are cross-linked. You have to remember the numbers, 20 to 30 microfilaments, also known as actin filaments. You know, there are two types of microfilaments, actin and myosin, which are cross-linked regular intervals by actin binding proteins, that is fascin and fibrin. The microfilaments are anchored to plasma membrane at top and side of the microvillus. First, let's see over here. What does that line means actually? That line means that actin is binded to the another actin and to the another actin and to the another actin and to the another actin to form a structure which is cross-linked to each other. You can see this is fibrin, this is considered the yellow color one as fascin and blue color one as fibrin. They link together two types of two actins so that they bind together. Okay, now coming up to the different one. At top and side of the microvillus, microfilament extend down into the apical cytoplasm where they become embedded in the terminal web. Okay, within the terminal web, the actin filament becomes associated with the molecules of spectrin. Now you see over here, we were binding the actin with fibrin and fascin, but in the terminal web, the actin filaments are associated with spectrin, not with fascin or fibrin. because the actin filaments are associating with myosin 2 this time, not the actin filaments to the other actin filaments. The columnar epithelial cells lining the luminal surface. Okay, so this is the example of the small intestine bears the numerous microvilli on the free surface to be vertically striated and is referred as, see over here, these are vertical striation. First, let's consider that what was written over here. There was written that on the apical surface the microvilli extend downwards. Wait, let me change the pen. Extend downwards. See here they are extending, and over here they get associated with the apical surface. Where they get associated, they form the terminal web. See over here, you can see the above structure is different from the below structure. See, there are different striations going on over here. So this is the form which is known as terminal web. Here you can see it's mentioned terminal web. Now we will see that myosin 2 are associated with actin. Here you go. This is myosin 2. This is myosin 2. This one right here. And they are associated with actin. Where they are getting associated, they are linked by the spectrin protein. You can see this spectrin proteins are present over here. Here you go. There you go. This is the spectrin linking the myosin with actin. Now you'll get it that it is myosin 2 type, not myosin 1. You know, myosin 2 is mostly considered. Now see, vertical striation, what do you mean by this? If the villi are present like this, the luminal surface of the cells lining the proximal or convoluted tubules of the kidney, they are longer and appeared as brush border. Luminal surface of the cells lining the small intestine appears to be vertically striated and is referred as striated borders. They are small than the they are smaller than the microvilli present at the luminal surface of the proximal convoluted tubules of kidney. It's mentioned over here. The proximal convoluted tubule of kidney contains larger villi. You can see over here. There appears to be brush borders. Remember that. What's the brush border and what's the striated border? This one here is known as striated border. Striated border as they are small. And this one right here is known as brush border as they are larger and they are present in the proximal convoluted tubule, while the striated border are present at the luminal surface of the small intestine. That's the function of the microvilli. We have already discussed that they are immovable, that is, they are immobile, but functions to increase the surface area to increase absorption. Now coming back to cilia, we have discussed there are two types of cilia, but we haven't discussed the difference. Why are they characterized as kinocilia and stereocilia? Cilia are actually hair-like projections, while we have discussed microvilli was like finger-like projection. They are hair-like extensions of the apical cytoplasm, we can say that in simpler words. While the description mentioned over here is as cilia are hair-like processes found chiefly on the free surface of the epithelial cells, which are specialized for the transport of fluid. Yes, that's the most important point. Fluid or a film of mucus over the epithelial surface. So here it's mentioned the difference of kinocilia. The kinocilia are motile, that means they can move the mucus layer from the upper epithelial cells, while stereocilia are non-motile, so they cannot. Then why are they considered as cilia? What is the importance? First discuss our location. The location is that they are present in the kinocilia are present in the pseudostratified columnar epithelial cells of the conducting part of the respiratory tract. We have discussed it in the pseudostratified columnar epithelial lecture, and that is the same lecture, and the conducting part of the respiratory tract, and simple columnar cells of the uterine tube, and neuroepithelial cells of the internal ears. Have you noticed something over here, that this type of modification is present on the columnar cells? They are present on the, that is, one type is pseudostratified columnar cells and the other type is simple columnar cells. Okay, remember that they are found on the stratified or non-stratified columnar cells. Now coming to our, you can say, structure of cilia. So we have discussed this, that the structure of microvilli contains microfilaments, while over here you will see that the structure of the cilia contains microtubules. That's the difference. Okay, so considering this as our, the structure in our yellow is our cilia. So in a transverse section from the upper surface of the cilia, and we have seen that they contains small, you can say, pairs of microtubules surrounding pair, which is actually not a pair, it's lying in an individual form. So we'll call it as central sheet, as it's different from others and it's present at the center. So this whole structure is known as axonium, which is named in like Hussain. So we will discuss the what is axonium. Axonium is central structural core of cilia and flagella, composed of microtubules. We have discussed that in the microvilli we have microfilaments, that is actin and myosin, while in the axonium we have, or the cilia, we have microtubules arranged in a characteristic pattern called 9+2 pattern. We will discuss why this is known as 9+2 pattern. 9 peripheral microtubule doublets surrounding 2 central single microtubules. This structure provides the necessary support and flexibility for ciliary and flagellar movement. Now considering at that line 9+2 pattern. Now count these dots in green, that is pair of dots, or the pair of microtubules in a simple way. So 1, 2, 3, 4, 5, 6, 7, 8, 9. This nine these 9 pairs of microtubules forming a ring-like structure with the central core that is having one individual. You can see there are no linkage between these two microtubules, so we will consider them as two individual microtubules. That's why it is known as 9+2 pattern. Now you get it? 9 that surrounds the 2, that is 2 individual. Why don't we call it 9+1+1? Why? Because they are already getting surrounded by the central sheet and is considered to be as a single structure. That's why 9+2. Okay. Now if we take the cross section from the lower part of the, you can say, transverse section of the lower part of the cilia, that is from where they arise. Lower part, like the terminal web, we saw that in the cilia we have the terminal web and it contains actin and myosin too, while the structure that was elongated, which was actually called as the microvilli, was having actin binded to the actin, while in the terminal web the actin was binded to the myosin too. Okay, now over here the basal surface, we don't call it as terminal web, we call it as basal surface of the cilia, contains some difference in their structure. What is the difference? We have seen in the axonium there are 2 pairs of microtubules, while over here we can see that there are 3 pairs of microtubules. Okay, so we'll see that when they extend, I can show you this one. Consider this as one. When they get extended, they are consider them that as the 2 long microtubules. These are the 2 long microtubules and this one is the 1 short microtubule that remains back in the basal surface instead of going to the axonium, that is the uppermost part having the 2 microtubules. Okay. Functions of cilia, we all know, we have also discussed that they give effective stroke, which is the fast forward movement which moves the cilia in the forward direction. They give forward bend and they help in the mucus movement from the epithelial cells. And there is an recovery stroke. The recovery stroke is not that much fast, it's only for the cilia to regain their own original position. Okay, this movement allows the mucus movement and also allows the ovum to get into the uterus. Mucociliary transport or mucociliary clearance, a protective process that ensures the removal of pathogens before they can reach the delicate and vulnerable tissues of lungs. Now you get it? They help in the movement of the mucus so that the mucus do not stores the pathogen and does not causes the infection in our respiratory tract. Kinocilia on neuroepithelial cells act as a receptors. Now, so this is the most important point, that why are kinocilia present in the neuroepithelial cells? I haven't discussed this before. This is because of the fact that this one is not included in the movement of mucus or any other you can say, not in the movement of ovum or something. They act as a receptor. They carry the reception of the balance. They help in the maintenance of balance. They do not, that air is present in our body. They help to maintain the body balance. They stop the person to fall on its one side. So they are also the most important. They also serve as the most important function. Now coming to the ciliary dyskinesia, primary ciliary dyskinesia, which is the clinical application of the cilia, also known as immotile cilia syndrome or Cartaginer syndrome. It is a rare autosomal recessive disorder characterized by the defect in action of cilia. Okay, these are our cilia, and they help in the when they move. They help in the movement of the mucus. They act as a balance neuroepithelial cells, balance receptors, and in the fallopian tubes they act to serve the ovum to the uterus. What if these all these functions get disturbed? How do they get disturbed? They get disturbed by the movement, because of the no movement, that is the cilia becomes immotile. Why cilia becomes immotile? Due to the genetical changes. So what will happen to the person? The person will most likely be sterile, because there will be no movement of sperm, as we have discussed that these are ciliary movement. Cilia has ciliary movement and flagellary movement, so no movement of sperm, and the ovum will not reach the uterus because the cilia are not motile. Then coming to the mucus get deposited on the you can say respiratory tract, and it will lead to certain different problems. Now, primary cilia. It is non-motile, but structure represents that of the kinocilia and serves as antennae that enables the cells to gather information about their movement. It's not that much important. Now coming to the stereocilia. Stereocilia resembles cilia, but because of their immortality, they were named as stereocilia. Now you get it why they are known as stereocilia and why not kinocilia, because stereocilia are immotile and kinocilia are motile. Now coming to the lateral domain, which is present on the side of the epithelial cells. We have discussed it before. We have already visualized it. I have already shown you the picture so that you can easily go through and cope up with the details we are going through it. So the lateral domain of each epithelial cells lies in close contact with the lateral domain of other functions. Now I have to show this to you. Consider this as our one epithelial cells, that bump over there, that I have made that so that you can recognize too. So just let me change it. Okay, consider this as your two epithelial cells. So we have discussed that this one is your lateral epithelium, this one is your lateral epithelium, this one is your lateral epithelium. The thing with the statement says that the lateral domain of one epithelium comes in close contact with the lateral domain of other epithelial cells. So this is the thing that was written. You all know when the cells combine together, they will come in close contact with each other. That is, the lateral domain will be in close contact. So in order to identify the lateral domain, we have to see the presence of adherence. I have told you that we will discuss in another chapter that these are cell junctions. Then that they are present in cell junctions. Presence of invagination and evagination. Presence of adhering and occluding junctions. This protein is present in cell junction, and these are cell junctions. Cell to cell communication by gap junction, that is, cell comes in contact with other cells. These cells come in contact at particular region. Above this, this region is already bind tightly to the other cells, like no exchange will happen over here or down there. Only exchange will happen over this region, which is known as gap junction. Okay, now coming to the basal domain, the lowermost part of the epithelial cells, they contains basement membrane. Beneath the basal domain is present our basement membrane, and beneath this basement membrane we have that connective tissue. I have told you this, these are connective tissue. Consider them as your connective tissue. And then on the basal domain we have hemidesmosomes. Consider them as your hemidesmosome. These are also a specialized type of the cell junctions. We will discuss in the cell junctions topic. And enfoldings of plasma lemma. You have discussed that there are enfoldings present in the apical surface, and now we will be discussing that there are enfoldings present in the basal domain also. Why is that? So these enfoldings are not modified, but they are present over there. There are small enfoldings. You can see over here they are present. They are present over here. They are present. And let me go through hemidesmosome. See over here, this is the hemidesmosome anchoring junction. Basement membrane, also called basal lamina, thin layer of acellular material. Now we will be discussing basal membrane, not the basal domain. They are two different things. Remember that the basal domain, basement membrane are two different things. So remember the fact that they are acellular materials between the epithelial cells and the underlying connective tissues. Okay, so the basement membrane is further divided into basal lamina and reticular lamina. So basal lamina. So the basal lamina are fibrillar layer lying directly beneath the basal surface of the epithelial cells. Due to this reason, we call basement membrane as basal lamina also. Make that clear that these are three different terms: basal domain, basement membrane, and basal lamina. First, get it clarified. Basal domain is actually the lowermost part of the base of the epithelial cells. Basement membrane is actually the acellular region between the epithelial cells and the connective tissue. And now we will come up with the basal lamina. Basal lamina is the part of the basement membrane which is in close proximity with the, what do you call it, epithelial cell. That's why the basement membrane is also known as basal lamina. Although there are two types, that is basal lamina and is reticular lamina. Two layers, that is basal lamina and reticular lamina. They are composed of collagen type 4, blah blah blah, glycoprotein, laminin held together. You have to remember this as your MCQs, nothing to be explained. Then they are further classified as two layers, that is basal lamina also contains two layers, that is lamina lucida and lamina densa. Lamina lucida lies just under the basal surface. See, now I have to show you that. Consider this densa will be, this is your lamina densa, and this whole is your basal lamina only. This is your basal lamina, and this is your basement membrane, and that the basal domain will be above it. This one would be your basal domain. Remember this, these are two confusing terms. Now the lamina densa contains collagen type 4, fibril, laminin and perlequin. You have to remember this, these names. Okay, now reticular lamina, below this will come our reticular lamina. Let me change the color and improve the thickness. This would be your reticular, this would be your reticular lamina. Now see what's written in reticular lamina. Layer of variable thickness contains reticular fibers, gags, and gags are glycosa aminoglycans. You will discuss this in biochemistry. Chondroitin sulfate, these are types of gags actually: chondroitin sulfate, heparin sulfate and hyaluronic acid. You must have discussed this and in the what do you call it, carbohydrates chapter in the polysaccharides. Okay, and collagen type 7, you will discuss this in biochemistry. Biochemistry will study that type 4 collagen and type 7 collagen are usually found in the, or you can say limited to the basement membrane, to underlying connective tissue. It is considered to be the part of the underlying connective tissue. So as you all know, after the basement membrane we have that connective tissue layer. This connective tissue layer, this connective tissue layer, this connective, as they are in close proximity with the connective tissue, and the basal lamina are in close proximity with the basement membrane. So the basal domain, or basal domain, no basal domain, my bad, basal lamina is considered to be the part of the basement membrane, while the reticular lamina, which is this one, is considered to be the part of the connective tissue. I hope that you all get this one, because it's contains two confusing terms. Coming to the fact that the basal domain contains hemidesmosomes, which are the junctions which we have discussed in the other lecture, that anchors the epithelial cells to the basal lamina. Let me show you this thing. See, these are our hemidesmosomes. This is our epithelial cell surface, and this is our basement membrane. This is our basal domain. So they are linking the epithelial cells to the basement membrane. Yes. Now the enfoldings are present, we have discussed. They contains they help in the active transport of ions and molecules. They contains mitochondria. You all know that active transport requires ATP. ATP is formed by the mitochondria, so mitochondria are present in a large amount in the enfoldings. So now the basal striation are most prominent in the epithelial cell lining, the proximal and distal convoluted tubules of the kidney. Functions, they bind the epithelial cells to the underlying connective tissue. Molecular sieve or ultrafilters, that is, they are semi-permeable, they allow certain substances to pass through them. Scaffolding during epithelial regeneration or wound healing, that is, what do you mean by scaffolding? That is, they act as a mold, you can say act as a mold, or you can say they support the wound healing or regeneration. Helps in cell growth, proliferation, differentiation, bioregulation, interaction between the cell surface receptors and the molecule in extracellular matrix. Functions of epithelium. In general, epithelia, which secretes, absorbs and filter, are single layer their height. Okay, we have discussed that in the simple squamous epithelium, that they absorbs while in the cuboidal, simple cuboidal, we have discussed that they secrete. This is the same fact written over here. What do you mean by single layered? Single layered means over here that they are simple epithelium. Squamous to columnar correlating with their efficiency. That's the same thing. The pseudostratified epithelium is generally engaged in protection, secretion, and ciliary transport. You get that? Because of the fact that pseudostratified and stratified epithelium are not single layered and they have many layers, so they are most usually involved in protection and secretion. Role of stratified epithelium is generally limited to protection. Protection, all type of covering epithelium provides protection to the underlying connective tissue, and this capability is directly proportional to the number of layers present in epithelium. You all know that more the number of layers are present, more would be the thing under it would be protected. For suppose you need to protect something, for suppose you need to protect your for me, I have to protect my stylus pen. So in order to protect it, I had cover over it. The thickness of the cover depends on the protection need. That means if the thickness is more, my stylus pen is more protected, but if the thickness is less, then the stylus pen is less protected. If it will fall, it's most likely to get damaged. Same goes for your screen protector. That's the most convenient example. Provides protection at the surface of the conjunctiva of the eyeball. The transitional epithelium, also known as urothelium, it's urothelium, it's my bad, it's urothelium. Remove this H. Urothelium performs the function of the protection of urinary passage, including the prevention of the reabsorption of urine. The stratified squamous oral cavity and in vagina furnish examples of protection against mechanical trauma. Okay, the stratified squamous keratinized epithelium, we have discussed that they protect against drying, soaking, and bacterial invasions. Secretion, the glandular epithelium helps in secretion, while in absorption, we have known that squamous epithelium allows absorption. You have to go through these whole paragraphs. It will extend my, you can say, my lecture. I've already done so much lambi on that. Okay, so that's urine. Urine and sweats are the example of such filtrates. Then in transport, you can say the cilia, the modification of the apical surface present helps in the transport, and the sensory receptors on the neuroepithelial cells as well. Coming back to the stereocilia, we need to discuss that what is stereocilia. We haven't discussed that in much detail, so I'm going to elaborate it more. So stereocilia, why they are not considered as kinocilia? First, most important reason is that they are not able to move. Okay, then the other fact is that they are usually long microvilli, as their structure appears to be similar to the macrovilli, but they are not considered to be microvilli because of the fact that they are longer and they contain bundle of microfilaments. They are considered as microvilli, although we know that cilia contains microtubules, not microfilaments. The other fact that they are not considered as microvilli is that they are twisted around each other. You are seeing over here that they are twisted around each other, giving that tuft-like appearance. Cells bearing stereocilia have a restricted distribution in the body. Mostly they are found in the duct of epididymis and vas deferens. In addition, the stereocilia are also found on the neuroepithelial cells of the internal ear. The stereocilia in the epididymis and in vas deferens functions to increase the mucosal surface area to facilitate the reabsorption, same as microvilli. The fluid that leaves the testes along with the spermatozoa. Function of stereocilia present on neuroepithelial cells that of the internal ear is quite different. They function in nerve signal generation. Deflection of stereocilia by head movement or by the sound waves causes depolarization of hair cells, plasma membrane, and thus the nerve impulse is generated, which is transmitted to the central nervous system by the sensory nerve axon. That is, they act as a mechanoreceptors in the ears. That is, they hear the sound waves and they carry the sound waves to the CNS in order to generate the signal that somebody is saying something, in order to recognize some voice or something like that. Now you get it? Okay, bye.